Good afternoon. So I'm going to talk about amphibians, which is pretty fun and exciting for me. All right, I would like to acknowledge the other authors. There's lots of people who've contributed to this research, both in terms of developing the protocols and implementing the study, field data collection, and statistical analyses. But the people I would like to acknowledge right now are the people who are also an author on this presentation, and that includes 3 others from Department of Fish and Wildlife, Timothy Quinn, Mark Hayes, and Eric Lund, as well as Jay Jones with Weyerhaeuser Company. And he's really been our statistical guru and go-to guy with statistical questions. And actually played an even larger role than that, but I won't go into details. All right, so the amphibians that we're talking about are forest practices, stream-associated amphibians. Again, there are 7 amphibians that are listed for protection under forests and fish, and those are here. We have 2 species of tailed frogs, coastal and Rocky Mountain, 3 species of Tournet salamanders, Olympic, Columbia, and Cascade, and then 2 species of lungless salamander, Van Dykes and Duns. And again, we've mentioned this several times, but to remind you, oops, I don't have that same thing in this presentation. The ones that we were looking at for a response in this study included the coastal tailed frog and the 3 species of Tournet salamanders. The primary reason for excluding Rocky Mountain tailed frog is that it is found only in a very small portion of the state in the Blue Mountains of southeastern Washington and largely outside of the lands that are managed under forest practices rules. And then the reason that we did not look at Van Dykes and Duns salamanders is that though they are stream associated, they do not have any in-stream phase to their life cycle. So they actually breed on land and lay their eggs on land, which means that the sampling protocol we would have needed to use to sample those amphibians is vastly different from the sampling protocol that we were implementing in-stream. And then we also have giant salamanders. Now, while they are not forests and fish or forest practices species, copes and coastal giant salamanders were a species that were found throughout all of our study sites. And further, the Cope's giant salamander is found in each site and was a good candidate for the genetics component of our study. So we're not going to talk about the genetics piece here today, but there were 2 species that we looked at genetics for a response, and that was the Cope's giant salamander and coastal tailed frog. So the study objectives that I'm going to present on here today for the 3 stream-associated amphibian taxa, so again, coastal tailed frog, the 3 species of Tournet salamanders, and the 2 species of giant salamanders, were to compare occupancy before and after harvest across our treatments, compare abundance before and after harvest across our treatments, as well as to investigate the presence of those amphibians in areas that were obstructed by large amounts of woody debris in the stream, largely in the form of slash and wind throw. So first I'll talk about occupancy. We evaluated amphibian occupancy with a light touch sampling method. This is a daytime survey that we conducted between about 7 a.m. and 7 p.m. between July and October of each sample year. We had 2 years of pre-treatment data collection in 2006, 6 and 7, and 2 years of post-harvest data collection in 2009 and 10. And it's a systematic sample of the stream, non-fish bearing stream length. So beginning at the fish, non-fish break, and then sampling systematically between that point all the way up to the head walls on every single stream tributary. So it's really looking at basin-wide abundance pretty effectively. And with that sample, you're basically moving up the stream channel network, regardless of whether or not there's water in the channel at that point, turning over any movable cover objects that are cobble-sized or larger within ordinary high water, looking for amphibians and capturing them as you find them. And so I'm just going to jump right into results. So results for the coastal tailed frog. We detected coastal tailed frog at 15 of our 17 study sites. Now, if you remember, presence of the amphibians was one of our criteria for selecting a site to begin with. So the fact that we didn't find this species at all of our study sites during the study period really gives you an idea of how ephemeral the detection of that occupancy can be. There were 950 detections in total over the 4 sample years, and the number of detections by site and year ranged anywhere between a single individual to 105 individuals for a single site and year. And the average number per site and year was 18. We detected a change in occupancy at 2 sites, and that was going from detecting the species to not in the pre-treatment or pre-harvest period to not detecting it in the post-harvest period for 1 site, and that site was a 0% treatment basin. And then we saw the opposite at another site where we did not detect the species in the pre-harvest period and then did detect it in the post-harvest period at one of our 100% treatment sites. For Tournet salamanders, we detected them across all study sites. This was by far our most abundant species in terms of numbers observed through time, with nearly 7,000 detections, and the number per site and year ranged anywhere from a single individual to 406 individuals in 1 study site, with an average of 107. We did see a change in detection of occupancy at 1 site, and that went from detecting it in the pre-harvest period to not detecting it in the post-harvest period, and that was for a forest practices treatment. And then for our giant salamanders, we again detected this species across all study sites. We saw nearly 2,400 animals detected across all study years, and the number per site and year ranged anywhere between 1 to 207 with an average of 36. And we did not observe any changes in detection across the study sites for the pre to post-harvest periods. So in summary, we did see only minor differences in pre and post-harvest occupancy, and that was for 2 of the 3 species, so only for the coastal tailed frog and the Tournet salamanders, and that was only in 3 of our study sites, 3 of the 17 sites where we saw a change in occupancy, and all of those sites were in our Olympic block. So, and then there was 2 negative responses where we went from seeing the animals to not seeing them in the post-harvest period, and then 1 positive response where we did not see them in the pre and did see them in the post-harvest period. So, do changes in pre versus post detection truly reflect changes in occupancy? That's the big question, right? We noted that for those 3 sites in the Olympic block where we did detect a change, there was pre-harvest variability in detection as well, meaning that for one of the 2 pre-treatment sample years, we did not detect the species in those sites. So there was already some very annual variability that we observed. And we also noticed that for those sites, there was comparatively few detections across all years, so 6 or fewer animals observed at those sites. So lower individual detection or abundance may contribute to non-detection of occupancy even when animals are present. So we hesitate to say that though at some sites we did not detect the species in the pre or post-harvest period, that they truly were not there, even though we feel like we very rigorously sampled for the animals. Our solution, one, is to do multiple pre and post-harvest sample years. In several instances, if we would have only done a single year of pre-treatment sampling, we would have not detected species where they did occur in the pre-treatment period as well. And then also to extend sampling through time. And that's why we're actually getting ready to conduct both the demographic and the genetic amphibian sampling in 7 and 8 years post-harvest sampling, and that will be initiated in July of this year. So that was number 1, was looking at occupancy pre versus post-harvest. Number 2 was looking at abundance pre versus post-harvest. Lots of folks have used a sampling method similar to our light touch sampling to index abundance of amphibians through time. And so why didn't we use just our light touch counts as an index of abundance? Well, confidently estimating true abundance does require estimates of detection, so the likelihood of detecting a single individual in the stream. And detection probability may vary as a function of covariates. And so our solution was to utilize a new tool for estimating detection. Specific influences on detection that we were concerned about included environmental variability. So this is a long-term study. We know that these stream-associated amphibians are susceptible to changes in the environment, like variable rain conditions or temperature conditions. And so we wanted to ensure that some sort of environmental change was not influencing our ability to detect individuals. And then also we were applying treatments. So we have this baseline data collection across sites, and then we're applying specific harvest treatments and then sampling those same sites again. And specifically what we were especially concerned with was that there could be a confound between between detection and treatment. Specifically, we've seen in other presentations that the amount of wood debris that ends up in a stream after harvest varies according to our treatment, with the more severe treatments that did not have any riparian buffers receiving the most amount of new wood input into the stream. And the more wood that you have in the stream, the less able we may be as samplers to apply our sampling methodology, that light touch method, in order to detect the species. And so we found out that we were right, which we already know from seeing the large woody debris and vegetation presentations. We did see a lot more wood in unbuffered reaches of our treatment streams. These are just a couple examples of what some conditions at a couple of sites looked like pre-harvest, and a very extreme example, just to give you that comparison, in the post-treatment period. This is a reach that's, this is a 0% buffer treatment, and it was also impacted by blowdown from that 2007 December storm. But this is a lot easier for us to navigate the stream channel and turn over those cover objects and look for amphibians than this. And so we wanted to make sure that we weren't, that there was no confound in our ability to sample the amphibians and the treatment. And so we were able to adjust our light touch counts for detection. We did not feel that it was in our best interest to use standard mark recapture techniques because of time and logistical constraints that would have made that very difficult to apply this in our study setting. And so we were presented with an opportunity actually to collaborate with Weyerhaeuser, and in specific with A.J. Kroll and Jay Jones, who helped us to utilize a recently developed binomial mixture model where you're actually able to go in and estimate both detection probability and abundance simultaneously without marking individuals. This was huge for us. And that method was developed by Royal in 2004. So this approach relies on spatially and temporally replicated counts of unmarked animals. That was one big key ticket for us, was that we weren't trying to track the same individuals through time with individual marks. There's a couple assumptions. One is the closure of the population during the sample period. So from the start of the sample to the end of the sample of a single plot, you're assuming that individuals are not moving in or out of that plot. And an independence of counts across sites was the second major assumption. The other advantage of this modeling that Royal developed is that you're able to include covariates in your detection models and actually in your abundance models as well. So anything that you think might be influencing abundance or detection, you can include that in the model to account for it. So specifically, we included a riparian stand condition, again, to address that concern that the woody debris loading may have been impacting our ability to detect the animals. We had 3 categories: an unharvested category, which would have been applied to our reference sites, and then a buffered category and an unbuffered category. So the buffered category would apply to our 100% treatment sites and the buffered reaches of the forest practices treatment, and then the unbuffered would apply to our 0% treatment sites and the unbuffered portion of our forest practices treatment. We also included stream order, stream temperature, and year as detection covariates for torrent and giant salamanders only. We would have liked to have included those also for the coastal tailed frog, but because of small sample sizes, we were not able to include those in the model. So again, this is the same light touch method, but it's applied to detection plots that are 30-meter detection plots that were systematically distributed across all study sites. So it was the same daytime sampling, the same turning over of surface cover objects within ordinary high water, but just restricted to these 30-meter detection plots. And then we revisited each plot on 3 occasions. And repeat samples were at least 1 day apart, such that we felt that 1 day would allow amphibians that had been handled to kind of readjust in the stream. And then also repeat samples were conducted by a different sample to reduce bias. So if I did samples 1, 2, and 3, I might become sort of familiar with where I saw that huge giant salamander in that pool over there on samples by the time I got to the 3rd sample. So that eliminates that concern. Okay, so then we were able to estimate detection probability for those detection plots and then adjust the basin-wide light touch counts for imperfect detection, including those covariates. And we were able to calculate a weighted average of adjusted counts based on the length of stream meeting those different criteria, the different covariate criteria. So this is just really quickly, we did detect an evidence of a covariate effect on detection. And we saw that the stand condition, which was that reference versus buffered versus unbuffered condition, did affect detection for tailed frog larvae, Tournet salamanders, and giant salamanders. We did not see that same evidence of an effect for tailed frog post-metamorphs. And we also detected evidence of a covariate effect for giant salamanders for stream order. Okay, and so now I'm just gonna jump right into the abundance results. This is on the log scale, so the scale is not so important. What's important is the bars and the dots, basically. So don't worry about the scale here. But we do have reference 100% forest practices and 0 treatment. These are the same for each figure here. Here we have tailed frog larvae, tailed frog post-metamorphs, Tournet salamanders, and giant salamanders. And what we're looking at is the change in the estimated abundance pre versus post for each of those treatments. So for example, here, we did see a small increase. Well, wait, let me go back 1 minute. So the dots are the estimated change from pre to post harvest, and then these are 95% confidence intervals. And no change would be any values that overlap the 0. A positive change in abundance would be anything that's above the line, and a negative change in abundance would be anything that's below the line. So my example then going back was that we did see a small increase in the reference abundance for tailed frog larvae. And then the p-value here is that period by treatment interaction term, which again is really our term of interest. And that's telling us if the difference, if the change that we observed was different by treatment, varied by treatment. And in this case, yes, it did. So again, this is tailed frog larvae, and we saw an increase in abundance in the 100% treatment and in the forest practices treatment relative to reference conditions. For tailed frog post-metamorphs, we saw an increased abundance in the 0% treatment relative to reference conditions. For Tournet salamanders, we observed no changes across any of the treatments. And for tailed frog post-metamorphs, we saw a decrease in abundance in the forest practices treatment. So that's it right now for abundance. And we're going to move on to presence and obstructed reaches. So like we've mentioned, there's these reaches that become highly obstructed by in-stream slash and woody debris in the post-harvest period. This is an example, again, an extreme example of that. The stream is right in here. This is the pip buffer at the top of one of those uppermost points of perennial flow. That's a 56-foot radius buffer that's required around the headwater, the absolute initiation point of the stream. And then here's the stream coming down. Pretty hard to get in there. So this is an area where we couldn't conduct the light touch at all. We could not use that sampling methodology to sample amphibians in those reaches. And previous research has often assumed that amphibians would not be in these reaches because it's just so clogged with litter needles that have fallen off the trees, fine sediment. It's just this matrix of muck down in there. And so there's this assumption that the amphibians wouldn't be there. Well, we decided to test it. And again, the concern is that there's this relationship between the amount of the stream length that has new wood and the severity of the treatment, so that we see more and more wood as less and less of the stream is buffered. And so our solution was to do these, the sampling in the obstructed reaches in these 3-meter long rubble rouse plots. And Eric talked about these plots earlier when he was presenting on the woody debris. And so it's the same plots. They cut down through, counted the sticks as they got them out. That was used for the large woody debris analysis. And then once we got everything removed, we were able to sample for amphibians in those plots. So there's 1 to 6 plots per stream, a total of 48 plots at 6 and 8 basins in 2009 and 2010 respectively. So again, these plots are only being placed where there's a ton of woody debris in the stream, which is in our more severe treatments. So we did not have these plots in the reference site, and we had most of these plots in the 0% treatment site. We placed block nets up and downstream to prevent amphibians from moving into or out of the plot, and then we remove all of the debris and stream substrate and capture the amphibians as encountered. And we're removing everything down to fines, 30 centimeters, or bedrock. And so here's very quick in a nutshell what the results are. And looking at this, really what I want you to take home from this graphic is that these are all of our sites down here on the bottom. And these are the number, they're not all of our sites, it's just the sites that we did the obstructed plots in. So 6 in 2009 and 8 in 2010. This is the number of plots that were placed in each of those sites for each of the year. And then this is just a total number of individuals for each of the 3 species, tailed frog, giant, and torrent, that were encountered across all of the plots in that year. The take-home message is that we did observe all of the species in those obstructed reaches, sometimes in very large numbers. So here we had 4 plots in one site in 1 year with almost 140 individuals detected across those 4 plots. That is a lot of amphibians in a very small 3-meter reach. So for tailed frogs, we did detect them in 2 of 6 and 3 of 8 of our basins in 2009 and 10 in these obstructed reaches, and the maximum number per plot was 8. For Tournet salamanders, we observed them in 5 of 6 and all 8 of our basins in 2009 and 2010, and the maximum number per plot was 60 individuals. That's 60 individuals in a 3-meter reach. Giant salamanders, we detected them in all 6 of the plots in 2009 and 6 of 8 in 2010, and the maximum number of plot was 19. The maximum number per plot was 19. So in conclusion, to summarize what I've tried to very briefly go through here today, for occupancy, we do not have any evidence that amphibians were extirpated from sites in the immediate 2 years post-harvest. For abundance, we did observe a negative response for 1 species, that was the giant salamander, and it was a negative response in the forest practices treatment where we observed an 82% decrease of what we would have anticipated based off of reference conditions. However, giant salamanders are, to remind you, are not a forest practices designated species. We did observe a positive response in 2 instances for tailed frog larvae in the 100% and forest practices treatments, where we observed 4 and 8 times the numbers of animals that we would have observed under reference conditions, respectively, and for tailed frog post-metamorphs in the 0% treatment, where we observed 6 times the number of individuals. And then also, we did not see a response or a difference in treatment for the Tournet salamanders. Amphibian use of obstructed reaches, we were very surprised at both the fact that we found all of the species in those obstructed reaches as frequently as we did, as well as what sometimes were in pretty vast numbers. So in conclusion, amphibians are not extirpated or immigrating out of those really obstructed /laden reaches immediately post-harvest. And if anything, it appears that there might be somewhat of a conglomeration of amphibians in some of those reaches. For detection, we did see that detection varied by the riparian stand condition for tailed frog larvae, giant salamanders, and torrent salamanders, and by stream order for giant salamanders. And our conclusion is that not accounting for detection could have resulted in biased results. I'd really like to stress the short-term nature of this study. We only have 2 years of post-treatment data that are described by these results, which would not describe the potential impacts to reproduction. So what we can say is that amphibians weren't immediately extirpated or like killed on site, nor did they immediately move out of the system or move into the system after harvests for any of the treatments. But understanding the long-term impacts would definitely require study over a longer temporal scale and over entire generational turnovers. So our recommendation is to continue monitoring after at least one generational turnover, and that monitoring should include both demographics and genetics. And as I mentioned, we are getting ready and setting the stage to initiate this sampling after a generational turnover starting this summer. So stay tuned, because to me, that's really, really a very important part of the question, is not were they still there 1 or 2 years after harvest, but what does that mean for them and their ability to maintain populations in those sites long term. And that's it. Thank you very much.